creo que vamos a iniciar. Buenos días a todos. Gracias por estar en el último seminario del año del programa de Frontiers in Genomics. Y bueno, pues vamos a cerrar con broche de oro. Hoy tenemos bueno, antes de presentar a Sergi, quisiera nada más recordarles que al final de la plática pueden alzar la mano aquí en el chat, en el Zoom, y pueden poner las preguntas por el chat, ¿sí? Y bueno, como saben, este es un programa que organizan el Centro de Ciencias Genómicas y el Instituto de Biotecnología, que está dentro de la licenciatura en Ciencias Genómicas. Bueno, como les decía, hoy cerramos con broche de oro este ciclo de seminarios de Frontiers in Genomics, y para mí es realmente un gran gusto presentar al doctor Sergi Castellano. El doctor Castellano hizo su doctorado en Experimental and Health Sciences en la Universidad Pompeu Fabra del Centro de Regulación Genómica en Barcelona, España. Después de ahí, hizo una estancia como Research Assistant en el Departamento de Biología Molecular y Celular de la Universidad de Hawái. Eso fue del 2004 al 2005. Desde 2005 al 2006 hizo un postdoctorado en también el Departamento de Biología Molecular y Genética en la Universidad de Cornell. Y después, desde 2006 al 2010, muy interesantemente, hizo otro postdoctorado en el Janelia Frown Research Campus del Howard Hughes Medical Institute. Después de ahí, en el 2010, se movió a Alemania para ser jefe de grupo en el Departamento de Genética Evolutiva del Max Planck Institute for Evolutionary Anthropology en Leipzig, que como ustedes saben es una institución muy importante en antropología molecular y la evolución del humano y en general de los primates. Y a partir del 2020 a la actualidad es profesor de genómica, genética y medicina genómica en el Institute of Child Health en el University College en Londres. Bueno, Sergi tiene un currículum muy amplio, entonces voy a resumir algunas cosas. Es una persona altamente ocupada, porque por ejemplo, también en la actualidad es el jefe académico de la sección de genómica del University College. También es el subdirector de un consorcio en el UK que se llama el GOC UK. Es el jefe de biología genómica y medicina de precisión, me imagino también en el University College, y también es el líder académico de la plataforma de ciencias genómicas y tecnología. Ha recibido muchos premios. Simplemente quiero destacar que recibió el Erasmus Scholarship del LEMBO para trabajar en bioinformática y genómica en el departamento de la Universidad de Manchester, y ha recibido muchos grants muy interesantes, ¿sí? Por ejemplo, quiero destacar algunos de los que tiene vigentes. Uno es referente a la terapia génica y los riesgos que implican, lo cual es muy interesante. También está desarrollando lo que ellos llaman el Single Cell Genomics, y de lo cual yo creo que nos va a platicar algo el día de hoy. Y muy interesantemente, con todo esto de la pandemia, ha sido el subdirector de un programa en genómica del COVID-19 en el Reino Unido, ¿sí? En el cual ha participado activamente. Y simplemente para seguir resumiendo, ha trabajado, tiene también un gran muy importante en el descubrimiento e interpretación de variaciones genéticas que tienen que ver con la deficiencia en micronutrientes en algunas enfermedades. Ha dado pláticas, lo han invitado a muchos lugares muy prestigiosos como el Cold Spring Harvard Laboratory, la Universidad de Harvard, entre otros, y tiene más de 11.500 citas. Algo muy interesante es que su trabajo ha sido destacado en muchas revistas, así con comentarios, así como cuando ustedes ven en News and Views de Nature, ahí ha sido comentado su trabajo, y también en periódicos como difusión científica, como en el Washington Post, The Guardian, The Times, The Economist, la BBC, El País, entre otros. Ahora bien, Sergi en general, para resumir algo de sus contribuciones, una de las más importantes ha sido en la paleogenómica y en particular en demostrar que hubo un flujo genético de los humanos modernos a los neandertals, también en la genómica evolutiva del chimpancé y los babuns, también en la evolución de las selenoproteínas y su participación en enfermedades en las que hay deficiencias en micronutrientes. Y bueno, pues el día de hoy nos va a hablar de Fighting Disease One Cell at a Time with Research and Automation. Bueno, Sergi, thank you very much to be here with us. I'm sure that we are going to enjoy your talk and we are ready.
Thank you very much for the kind introduction, Mario. Let me share my screen. Mm-hmm. Hold on a second.
Yeah, we forget to check that.
It should work, just not finding my presentation. Let me see now. There you go.
Okay, perfect.
And excellent. Thank you again for the kind introduction. As you said, I work in different topics, and I still work in some of them, like micronutrient deficiencies, and I've done a bit of ancient DNA work. But for the talk today, I thought I would discuss some of our more recent interests, and I'm gonna do that at the level of single cell work, because these are technologies that slowly are becoming more routine on the clinical side. And I'm gonna talk about single cell on the research side, but as you see in the title, I also added automation. And the reason for that is that in the clinical context that we try to use these approaches, automation is key in order to provide service to patients. So I'm going to try to tie up everything together and explain why we do things in a certain way and not others. So mainly I'm a UCL researcher, but we work very closely with the NHS, and the NHS is the National Health Service here at in the UK. Yeah, the hospitals, the doctors, and the clinical practitioners. As Mario mentioned, we also work very closely, and we are supported by the research side of the NHS in a way. So if we put all these together, at the end of the day, we are in a kind of proving ground in a specialized institute between the UCL and NHS, where we both have patients and research, because at the end of the day, what we are trying to do is to go a bit beyond the research and be able to translate our work directly into the NHS. That is always the goal. So some of the parameters that we think about are not necessarily what you think about when you are only focusing on research in your group. And these are, for example, among others, the speed. The work that we do has to work at a certain pace in the lab if we aim to translate into service into patients. Then throughput, the work that we do, the new analysis that we do, they need to be automated, hence the title, so that we can do it at a large volume. That is, for example, doing analysis for many, many different patients. And then, of course, the cost. The cost has to be very, very, very low. So we're moving a bit beyond of trying to plan a single experiment where we're trying to resolve a specific research question, where usually if it's a bit slow, that is fine, because probably we will do it only once or a few times. Maybe the volumes that we're trying to address are not that large, and maybe the cost is not the main worry if the funding it is available. But if we aim for our approaches to become routine in clinical care, in this case in the NHS, all these elements need to be taken into account. Now, I said that we need automation, and this is the reason that within UCL we have centralized all the automation, all the genomics services within this new building shared between NHS and ourselves. And this actually has been key for all the research and translation that I'm going to explain. The other reason I mentioned this is because I know that there are here many students of genomics, and job opportunities at core funded facilities, or as in our case, cost recovery facilities. And that means that we recover all the money that we generate. At the end of the day, we are free to the university, and I will talk a bit more on the business side at the end of the day. So this is a very legitimate work path for the future, and actually very interesting because there's an element of business there. Now, I said that I would mostly talk about single cell. This is why I said fighting disease one cell at a time. But more classic approaches are also interesting, especially because they have now been transformed translated into the NHS. So for many years in this environment, there's been a strong support into mutation discovery in patients with rare disease. And this has been generally successful here at UCL and at the hospital and in many other places in the UK and in the world, of course. This has led to very large research projects, sequencing, as it says here, 100,000 genomes, around 60,000 of them roughly from rare disease patients, around 30-40% of these patients coming from our hospital. And this sequencing has led, of course, to many diagnoses. So this technology is quite proven, scalable, and as I said, is fast, can do things at volume, and it's cheap. And this is why this has gone full circle, and all these approaches are now actually within the NHS. And this is the reason that some of us who are often more on the research side are doing less and less of this. In other words, these days, there's an NHS genomic service, which means that clinicians have the possibility, if they have a patient suspected to have a potential rare disease or some other condition where sequencing the genome would be helpful, they can do just that. And they will get not only the genome, but potential diagnosis based mostly on gene panels. So there's a scope for improvement. And this service was first applied to exomes, that is the coding part of the genome, the protein coding part of the genome, but has finally moved into genomes. So we've closed the circle. Now, of course, this is short read sequencing, Illumina sequencing. So there's scope for using other technologies like long read sequencing. And this is something that we're doing very heavily on the research side, hopefully, hoping that it will do exactly the same circle and getting finally into the patient. At that point is not in our hands, so we move towards other technologies that maybe appropriate for the future. Still, there are very specialized areas that we have contributed more, and that we are trying to contribute more in the future. For example, because we're in a very, very large pediatric hospital, it's very common to have potential rare disease patients in the ICU, and of course, these are many of them newborns with potentially fatal rare diseases. So we have this program where through genome sequencing, and we try to do this in hopefully less than a week, and potentially have a diagnosis that helps the clinician not only diagnose, but change, if needed, the treatment. And this is, of course, very useful for metabolic disease. And I thought I would mention this, which, although it's not my specialty, of course, at the time of the pandemic, when it started, we had just centralized all genomic services. So we had put also all the viral genomics at UCL together. And basically, we had all the robots and machines that allow us to contribute to a very large consortium across the UK of around 20 institutions, which had seed funding from the government to start large-scale sequencing. To be honest, at the time, and I was on a bit on the sidelines, because this is led by Judith Brewer at UCL, and I would just deputize for her when needed. It wasn't that clear how much this coronavirus would mutate, because some coronavirus mutate rather slow. So it wasn't so clear how useful sequencing would be. The fact is that the number of infections is so large that the virus has a very large ability to come up with new mutations, and some of those, of course, are worrying. At the end of the day, this consortium, which has now somewhat dissolved because the sequencing now has been passed into the NHS. And again, you see the goal, we do the research, we put everything in place, and then we shift it so that we can move to another research, has worked very well and has sequenced around 1,500,000 COVID genomes. That's around half of all the genomes that have been sequenced in the world. Our contribution from UCL is relatively small, and we mostly sequence them using long-read technologies. But we have here at UCL, Judith Brewer mostly has led other studies where basically we try to protect hospitals, trying to understand the patterns of infection by real-time sequencing. We can sequence COVID genome with short-read sequencing in less than 4 hours. And this has now jump-started other potential programs that we have submitted grants for working towards rapid infection discovery, not only of COVID, but of any potential pathogen, known and unknown. And that's also relevant because it is actually relatively often that you have infectious patients from unknown pathogens, and unbiased sequencing may lead to diagnosis of the actual pathogen, which of course is very useful clinical information. Now, finally, as I said, the main purpose of this talk is to discuss some of the potential technologies, single-cell technology in this case, that may eventually make it into the clinical setting as routine tests. And these are still, I would say, early days. These technologies are still relatively slow. They are in patients' numbers relatively low throughput, but most importantly, they are expensive. still there's a lot to be gained. And the reason for that is that disease is very, very heterogeneous. And being able to look at individual cell populations and individual cells, particularly rare cell populations, has become very, very important to understand not only the mechanics of disease, but potential diagnosis, potential prognosis, which allows, for example, to separate between healthy and diseased cells, which is relevant for many diseases and therapies. Also to use multi-omics approaches that allow us to characterize individual cells, not only from their gene expression, their transcriptome, but also from other technologies that I will mention. And then finally, these technologies produce data that is very amenable to artificial intelligence, machine learning approaches. And those are the ones that give us power to try to understand unbiased diagnosis, unbiased prognosis, and other uses that are helpful to the clinician. But as I said, we are early days here. So my goal for today was to present potential avenues of research that we're working on and how we're trying to push all these steps towards having the ability to diagnose disease and actually be able to predict how it progresses. So before going into patients and rare disease, we can take a bit of a step back and trying to understand disease itself and actually it is mechanisms. And of course, rare disease patients are by definition rare. Of individual rare diseases, there's so many of them that actually rare disease patients are common. But for individual diseases, sometimes is very powerful to have in vitro models of rare disease. And we can do that with two different approaches. One is using 2D cultures, and more recently, what we tend to call organoids or 3D cultures, where we have another dimension. But how well they represent rare disease patients is not so straightforward. So for example, something that we're doing is trying to understand these differences between 2D, 3D models of rare disease and actual samples from patients. I will not go in detail here, but at the end of the day, the expectation is that your models of rare disease should represent all the cell populations that you see in the actual patients, so that when you try to understand mechanisms or test potential therapies directly in vitro, the results are meaningful to what you should expect in patient samples. Now we can take this a bit further and directly study the differentiation process, comparing patient samples from controls, trying to understand if differentiation through development is not happening because of the disease. And just to give an example, we are doing this with Kabuki syndrome, which is a rare disease. We start with iPS cells and we differentiate them with either the mutation or without the mutation that leads to Kabuki, what we call histogenic lines. And we try to identify after differentiation whether they are cells that are only present in one of the two conditions. Okay. Now we can do this just with transcriptomics, but here is where multi-omics starts providing some power. For example, Kabuki syndrome is the result of mutations in genes which modulate expression, and at the end of the day, genome activity. So we can add to the snapshot that we take from the transcriptome in the same cell at the same time, also a measure of the genome activity of open chromatin. And this is what we call attack-seq. So we have two complementary views of each individual cell in patient samples and controls in the same genetic background. And the idea, of course, is that we will be able to understand what the mechanism of disease, and if we do so, we will open avenues to find potential treatments. So again, we're quite interested in understanding models of disease. And one basic question that has been addressed to some extent, but not fully, is how well differentiation works for these models, why some models fail to differentiate properly, and why some models differentiate with no problem. So we have a quite massive experiment, and we are reanalyzing this data. This is a very large number of cells, 800,000, and we can take samples at different intervals in the differentiation and try to ask whether genomic patterns can tell us why a line is failing and why a line is successful. In other words, what the measure that we are taking is whether the accumulation of deleterious variation, of deleterious mutation in vitro, and this is of course after mutations in the germline, after mutations are acquired in vivo, in the tissue, just from the moment that we take, in this case, skin, and then we develop these models and differentiate them, whether we can understand the mutations and the role they play in how well these models work. For example, we can see that the global number and pattern of mutations that are deleterious, that are accumulated, do not explain which lines will fail and which will not. It is mutations in specific genes that define the fate of the models that we are trying to create. And of course, we now kind of know the genes that are really, really important for these particular lines in neurodevelopment. Now, among other things, being a pediatric hospital, we're very interested in leukemia, which is often a rare disease in children. And for example, we're very interested in chronic myeloid leukemia, which is the result of a fusion gene between two chromosomes, what we call the Philadelphia chromosome, which is the fusion of a gene called PCR and another gene called ABL, which leads to having activation of tyrosine kinase pathways, leading to cancer progression and myeloid expansion that keeps growing till you end up in blast crisis. So this is a very tough disease, but there is a therapy for it because, as I said, there's activation of tyrosine kinase. So inhibitors lead to remission of cancer in most, but not all patients. So the power of single cell can be applied to these approaches because we can actually study patients at diagnosis, at remission after treatment, and afterwards when sometimes cancer comes back. And the reason for cancer coming back is because we have not removed all the cancer stem cells that eventually. So these kind of treatments tend to be for life. So a single cell, and in this case, with multiomics, can help us understand why some cells are resistant to treatment and which cells are not. So what I'm showing here is the separation of samples at diagnosis, remission, and then a control. And we are looking at cells that separate in these plots based on gene expression, but also based on the surface markers that they are showing outside their membranes. And this is based on panels of antibodies that we can also add at the single cell resolution. And then we have ways to identify which are stem cells and which are not. And for example, what we see, let me get the pointer, is samples of diagnosis. They have excess of stem cells that we know are cancer stem cells. They disappear after treatment for the most part, where of course control, they do not exist. You may ask yourself, how do we know that these are cancer cells? Well, among other things, and I'm not going to show this here, we can use long-read sequencing to try to read the fusion gene cell by cell, but we can also use the ratio of the BCR able in the transcriptome. And for example, here in red, this is the same image. The ratio here is such that this can only be in cells that have the fusion gene. Why is this relevant? Well, it means that we can actually quantify cells that are likely to be resistant to treatment. And actually, we can work with machine learning models that will be able to do that for us, given the data. So very early in the process, we can know how likely is that after treatment, the cancer will come back and how severe it will be. Not only that, because as I said, we are looking at the cell surface proteins in these cells, and we can separate cancer cells from healthy stem cells. We can also start looking for additional potential targets that can be used for cancer immunotherapy, which is one of the strengths here too. In this case, once you know that cancer cells uniquely express certain markers, what you can do, and this is done in the hospital for other childhood cancers, is to build chimeric T cells where one introduces receptors that uniquely will recognize those antigens that are expressed only in the cancer stem cells. So we are not only trying to understand how cells behave after treatment, but trying to identify new targets that would allow to kill those cells that are resistant to traditional treatment with kinase inhibitors. Now, this is an area that is particularly interesting to me, and more I'm geared toward this, which is trying to build new tissues with new properties that are useful for rare disease. There are many hundreds of rare skin diseases. Some of them are quite terrifying. And something that we started to do almost 3 years ago is try to see if we could accelerate skin cell culture. Remember that speed is one of the elements that is relevant to us. And actually, we can use certain inhibitors which help stem cells, push them towards differentiation and proliferation. And that seems to work quite well. But the problem is that we want to use these cells to build tissues and then skin grafts that we are going to use to replace, in this case, children's skin. So we need to be sure that by pushing the stem cells, we are not changing the properties of the tissues of the skin that we're trying to build. Otherwise, that could lead to other problems downstream. And one way to do that is yet again with single cell. And this is something that actually we've done, has worked quite well. We can show that the structure of the skin that is grown with or without our inhibitor is actually very similar. It's just that we grow the skin 5 times faster, that the proportion of the populations in the skin is the same after we discontinue the treatment for a few days before grafting. And then I will not go in detail here that many other of the properties that are relevant to us are not modified, so that when we graft the skin into the patients, we are grafting skin that has the structure and the properties and the necessary stem cells to self-renew, hopefully forever, so that the patient is cured. And as I said, if we can do this process 5 times faster than is possible today, it's not only easier to treat each patient, but also to treat more patients, and also to spend less time in the culture room, not saturate the culture room, which can then be used to other projects and also by other groups, because otherwise growing skin takes too many months. Now, another project that is growing on me a lot is something that is done only here and at Duke University, which is to, in a way, regenerate T-cell maturation in children who have been born without a functional thymus. And of course, the thymus is very, very important for T-cell maturation. And we don't fully understand what are the consequences of this transplantation. So this is another very clinically oriented project where single cell, we hope, can help us understand how the T-cell reconstitution is happening and how we can improve the therapeutic outcomes of it. Interestingly, the thymus come from donors that are having heart surgery, and then the thymus is not put in their chest, is put on their quadriceps, on their legs. But the maturation of T cells happens anyway. So this is very, very, very promising. Also related to regenerative medicine, of course, there are many rare cell populations that are very interesting to characterize with the idea to assess potential of regenerative medicine, and we're doing that. And now there are also very, very interesting technologies that unfortunately are still too expensive to move on to the clinical service side, but for the research side is very, very important to try to understand the spatial context, the local context, the cell communication of tissues and cell populations of interest. And we can do that with spatial transcriptomics, where we are not looking at individual cells in a solution, but at actual slices of tissue. And then we know the gene expression of all the cells in this tissue. Spatial transcriptomics at this point, at least the commercial kits that we use, is not fully single cell, but it is almost single cell. So we're also doing this for very severe preeclampsia, which of course happens to 1-2% of expecting moms. It has no treatment or cure beyond delivery and removing the placenta from the mom. And there's a few potential markers of disease early on. So we sample placentas from moms in delivery from 25 weeks, which is of course very early, to 35 weeks, which is late. Early and late preeclampsia have some differences. That's why we take these two time points. And we do single cell. And of course, because there is migration of certain cells, and there's communication between maternal and fetal cells in the maternal interface, we use these new techniques of spatial transcriptomics, so we can actually see the crosstalk, and in particular, the immune crosstalk, comparing cases, early cases to early controls, to late cases to late controls. And this is very much ongoing, but going well. Now, another aspect, and I think Mario mentioned actually this grant of assessing genomic therapy risks. And this is also a very interesting area. In particular, everything that has to do with gene therapy or genome editing with CRISPR or other technologies. So we are moving more and more with experimental and computational approaches to work on this. As you know, viral vectors tend to integrate, some more than others. AAV base vectors integrate less, lentivirus vectors integrate more, and they may integrate fully, partially, or rearrange, and may end up in the genome. And as you know, in the early 2000s, and actually some of the work was done here in the hospital, some of the early viral vector therapies and approaches unfortunately led to cancer in patients, and genomic risk since then has become a priority. So the approach that we are taking, and we've been doing this for a while, and others have done the same, is that we can build probes encompassing all the viral vector, and we can then take samples from the patient that is undergoing viral therapy or animal models, in this case, macaques, for example, and we can actually identify all viral vector integrations in a genome by looking at potential reads, potential sequences that span the viral host junction. That is, that they have part viral vector, part host, which can be human or can be mouse or other animals. Now, the question is how well this works. How good are we at identifying these viral integrations so that we can tell the clinicians, yes, your approach worked. There's no integration in any gene that we think may be oncogenic. We think you're not disrupting anything. So you should move on. And this is very important, of course, in preclinical trials before moving further. What I'm showing here, this is based on computational simulations. This is very interesting. This is what we do here, is that we take different vectors. I'm showing 4 here. They have different lengths. And then we simulate their integration into different chromosomes. In this case, these are human chromosomes. And then we assess our pipelines, how good they are in terms of finding all the integrations and not predicting integrations that do not exist there. At the end of the day, what we're seeing is that we can find, depending on the vector, between 80, 95% of all the integrations, that between around 80% of all the integrations that we predict are real. And then we have ways looking at less quality data to improve a little bit on that. So this is how you would measure pipeline. Then when you do this in real data, because the simulations are actually, I would say, quite realistic, quite well done, we can translate this expectation to the clinical side. Now, I mentioned that a lot of this is possible because we work in a context that we have these genomic services. And I thought I would give a snapshot for those of you that are thinking that core facilities can be a way forward. We are at the point at science that many of the problems that we approach, not only on the clinical side, but at any level, are not defined by the ideas, but by your ability to have the throughput to work on these ideas. This is why genomic core facilities are so relevant. And the one that we have at UCL is very dedicated, is around 10, 12 people. This is not my day-to-day, but I'm the academic lead. And basically what we do, we have all that set up, and then we cover all the costs. And of course, these machines are very expensive. So slowly over time, we add all the additional technologies. We obtain new funding to add new machines. And then there's a facility fee that when somebody wants to do something, helps cover all the costs and all the salaries. So for example, and this actually has grown quite a lot because COVID has added a lot of volume and money to this, but I think I have not updated these numbers in a while. Basically in green, this is the money that we use to pay all the salaries, the money that we use to cover all the equipment maintenance per year. You have to think that every time you buy a piece of equipment, some of them are quite expensive, every year you have to recover 10, more likely 20% of the cost of the machine for the maintenance cost. And this is every year. So you have to be quite ruthless with the cost to make it happen. Now, how you structure this? Well, you have people who are specialized in different technologies that they help each other. Then you need facility managers that coordinate all these. They are on top. They make sure that everything happens. And then you have academic leads that II's main job is to find money and think what is coming next and what we're going to do. And now we are trying to revamp all the bioinformatics service and actually generate money. And the numbers that I'm giving here actually is what is reasonable to recover as a cost, as a facility. That is, there's a lot of work that people do that does not bring money to the facility. That's, for example, training. That's, for example, often admin, taking care of maintenance of machines. If you are not performing experiments or analysis, you are not bringing money. And that's usually not taken into account. So these numbers, which are reasonable, is not what we do. What we do now is recover 100% of everything, which I'm hoping to move into a more sustainable model, because otherwise people tend to be overworked most of the time. And then bioinformatics analysis is not straightforward to make it cost recovery, because it's not straightforward to have an SOP knowing what is going to happen, because data is different. There's an element of research that, so we are trying to revamp all of these and hopefully getting into a more sustainable model for the future. For example, COVID was a very big lesson, because we were between closing the facility and then you cannot pay these 10, 12 salaries, or getting into COVID research as we did in order to maintain everything going. So cost recovery is doable, is cheap for the university, but it comes with their own challenges. It is fun to make it work though. So let me just acknowledgements to many of the members of my group, then UCL Genomics members who are extremely professional, and then many collaborators that are helping us to push some of the boundaries in translational research. And then I'm happy to take questions.
Okay, thank you very much, Sergi. A very nice talk, very interesting. So you give us a very nice overview of many applications of genomics in modern medicine. So now the seminar is open for questions. As I said, you can just raise your hand or you can put the question in the chat, as you prefer. Well, in the meantime, I have a question. The cases is spatial transcriptomics. You've had just one sample from one individual, and you can do spatial transcriptomics just with that particular sample of tissue or something like that. And how you compare, or how you know how reproducible is the result?
So like any other experimental design, you have biological replicates, which in this case would be different patients. For example, you would take 3 skin samples if you are interested in skin, 3 placentas if you are interested in placentas. Still, this is a good question, because there's still the methods. So when you do experimental design, of course, you do power calculations. Mm-hmm. And we know how to do power calculations to compare RNA-seq between samples. We kind of know now how to do power calculations for the number of samples for classic single-cell analysis of the transcriptomes. Spatial, it is still here, I mean, because it's very expensive, you try to go for at least 3 biological replicates, mm-hmm. preferably 6, but I'm not sure, at least to my knowledge, that the power calculations for spatial transcriptomics are really there, because everything depends on the size of the effect that you're trying to find in the comparisons. Mm-hmm. I have not been able to wrap my head around this. I was talking recently to 10x, and I don't think they have a very good answer to that either. Okay,
so as you mentioned, there are many possibilities with the application of sequencing, genome sequencing.
12 horas.
I'm sorry. And in your view, how you visualize the future? I mean, when is all these techniques are going to be able to be routinary in some hospitals in general? Because as you said, the techniques are very expensive, and you need to have a very highly qualified individuals to perform all this, with, I suppose, should have good salaries and everything.
Yes, it's good to try to pay people their worth. So we know that short sequencing has become routine. Long-read sequencing will soon become routine. And now the same project that I mentioned of 100,000 genomes with short sequencing, they are switching very quickly into long-read sequencing for the future. So what we are doing on the research side and many others, they will do it at a scale and would fit into the National Public Service. Now, sequencing is relatively easy. It's when you move into the more functional side that it is hard to scale. For example, RNA-seq is not so straightforward because the quality of the sample is much more determinant of the results. And many RNA-seq samples, when you do QC, they fail. It's not as easy. Genomes, of course, they are hard in the technology, but it has been relatively easy to scale. Now, with single cell, we are in a similar position, but it's even a bit worse. So that's going to take some effort. Is that usually you want to do fresh tissues. That means that you have to go from sampling to processing in the machine in half an hour. That's the ideal. That's the ideal time. So, and that's very different with the genome, right? That you can store your library and wait till you have time to run it. And so the pace is very, very different. So that's going to take quite a massive coordination, and the service needs to be structured in a different way. So I'm hoping single cell, at least that's kind of what we promise in grants. In 5 years, there will be certain tests or assays that will be done at the patient level in clinical labs. We work very closely with the clinical lab, and we have shared funding, but we are not. So what we do is start in the translational path early, try to push things, but always the goal is that they end up using all of this. And the jump of single cell, I think some of the simplest things may be there in 5 years, or perhaps there are other places in the world that they are already more advanced or more confident, and maybe in a couple of years there. We sometimes what happens now is that clinicians, for example, we had this patient at the hospital, and the clinician, it wasn't clear if this young lady, 13 years old, had chronic myeloid leukemia or acute myeloid leukemia. And of course, the treatment, it is different. So we have used single cell to try to look at that. And that is not routine, okay? But once you have streamlined the processing, you start seeing that may be amenable to a clinical lab in a few years in the future, where those cases where diagnosis is not straightforward, there may be ways to just do typical single cell and actually have a good understanding of the diagnosis, which sometimes is mixed between these two leukemias. So it stands to reason that there may be confusion at the molecular level.
Okay, there is a question from Michelle Moura in the chat.
I'm seeing it now.
Do you want me to read it?
Yes. Yeah, no, he's asking what is the best way to approach splicing errors. So I would say that it is bulk RNA-seq with quite deep sequencing, so you can see all the isoforms, and you can actually find the isoforms that may be losing or gaining potential exons due to splice sites changing. Now, the issue with this is that depending on the rare disease, you are not necessarily working with with the right tissue. And usually, of course, skin or muscle are the two tissues that we use for RNA-seq. But depending on the disease, the expression of the genes that actually have this splicing error may be low in the tissues that are easy to collect. We're actually looking for this kind of errors in patients in the ICU, for which genomic sequencing, whole genome sequencing, has not provided a diagnosis. These patients, unfortunately, they have all died, but identifying the potential splice error or isoform error is a way to provide closure to the families, where you can go back and tell them that this is what was underlying the disease of their children that unfortunately died very, very young at the ICU because there was no diagnosis and treatment wasn't clear.
Okay, no, there are no further questions, but I have another one. It's quite simple, but you mentioned you are doing a attack-seq with a single cell level. So, and can you do it? Can you do something like chip-seq at the same time? A single cell level, because sometimes with attack-seq you can visualize the open regions, the chromatin open regions. And many are going to be enhancers that are going to be working or not working. So, and the question is because not all the open enhancers are active, not necessarily are active, the ones that. So it's why yes. I'm asking about the chip-seq, because with the chip-seq you can use some tools to corroborate or to be more, corroborate the results with attack-seq, for instance, and to visualize which ones are active.
I would say it's likely to be possible, and possibly it has been done, but not necessarily in the commercial platforms that we use. These particular kits, they work with nuclei, not whole cells. So you can do the attack-seq and also the transcriptome. One issue I didn't mention is that, of course, when you reduce yourself to the nuclei, you have a big drop in transcripts. So the view that you get from the transcriptome is not as complete as you would like. Usually have 20-40% drop in the counts of transcripts, but you do have the attack-seq. So because you have, of course, the active regions of the genome and the transcripts, you may be able to link them in a way that, you know, okay, this is active, but it's producing nothing. This enhancer is activating this, but there's no transcript. So that may help a little bit. I think it's very likely that you can do actually any of the encode techniques at single cell level, but how well they work with all the other elements of, in this case, the 10x platform, that is a bit more complicated. For example, something that we're doing a bit with 10x, because it's not clear how well it works, is we are doing long-read sequencing for targeted long-read sequencing on single cells, because we're interested in these gene fusions. And it kind of works, but is not like commercially approved or anything. It's not that there's a kit that.
Okay, yeah, I understand. I see. So Laura Hinojosa.
Yes, thank you. So, well, for obvious reasons, a lot of money and a lot of resources have gone into mRNA vaccine platforms in the last couple of years. And how do you think these improvements will affect the applicability of similar approaches or of the use of mRNA platforms for gene therapy and precision medicine in these types of diseases that we talked about today?
So I'm definitely not an expert on mRNA vaccines or on RNA, but of course it seems what is more interesting to me is that the delivery system, the non-viral delivery system, seems to work very, very, very well, and that for gene therapy, it is very interesting, although it doesn't necessarily have like the specificity that some viruses give you for particular tissues where you're trying to correct a particular rare disease, for example, in the liver. So yeah, I would really need to think about that. I do need to read more about technology that has made possible all the mRNA vaccines. We were before discussing how viral vector-based vaccines and mRNA vaccines may be promoting different parts of the immune system. So yeah, I don't feel that confident to answer that question. Sorry.
Okay. Well, if there are no more questions, well, Sergi, thank you very much. Very nice.
Thanks to you.
Presentation, very nice talk. We now have a different view, not a different, but a more complete view of the approaches in genomics and medicine. So, bueno, muchas gracias. La próxima vez esperemos que nos visites acá. Y
muchas gracias.
Y fue realmente muy bueno que pudieras participar. Y bueno, le doy las gracias a todos. No hay más preguntas. Le doy las gracias a todos por asistir al seminario y estamos en contacto.
Gracias.
Gracias, Sergi.
Adiós.
Te volveremos a contactar, seguro, ¿eh?
Vale, perfecto. Adiós.
Gracias. Gracias a todos.
Gracias.